you guys want, I just want to fill in what we did the other day. So you got, I figured I could do it, or you can help me. So we had a list going based on categories. We had nucleating proteins, right? So for actin, what did we have? Yeah. Oh, that's hard for you guys on that side. Monomer binding. What did we? Oh wait, no. For microtubules. Gamma Turk. For monomer binding, what do we have for actin? And for tubulin, what do we have? Daphne. Okay, for severing, this is where we ended. For severing microtubules, Katanin, and for actin, Gilstolin. Okay, so that's where we ended. Good. Okay, business. Exam 3, I have made the executive decision to move to the 20th. I'm sorry I keep doing this to you, but I counted it and we have like 120 slides or something like that to get through, and there's no way I can do that in 2 days. Uh-oh. It's all blank slides. There's no content at all. Mostly movies and some drawings. So sorry if that inconveniences you. You're welcome if that's a welcome change. Okay, so lecture 17, I'm sorry for you guys who were counting on it, but the good news is you can listen to last year's lecture, and a student in this class has gone to the trouble to identifying where, and that is 23-24. I have not confirmed that, but I trust him. Okay. Yay for last year's recordings. Okay, so far this is what we've done. So we've got our key players, actin, microtubules, and our intermediate filaments, and then all the accessory proteins which regulate them. We've gone through our major concepts of dynamic instability, the fact that filaments are produced from subunits, nucleation, critical concentration, the fact that there's polarity, plus and minus ends for actin and microtubules, and treadmilling. We spoke about just the makeup of microtubules and actin, the fact that they have some sort of NTP bound, and the NTP is hydrolyzed upon addition. And then, of course, something I didn't put up here, but is the fact that the D-bound state lines more weakly, which is why the D-bound state is more apt to dissociate. We talked about different toxins that affect actin and microtubules, and you should think about how they might be useful in the lab or clinic. We spoke about homologs and bacteria, just for fun. And then we started talking about regulation of these cytoskeletal filaments with all these accessory proteins, and I'm keeping the same list on this so far slide as I'm creating up there. So we've got our nucleating, our binds monomer, our severing proteins so far. And then this week in recitation, you're going to kind of draw this monster picture together and figure out where each thing goes and what processes are affected by what and all that. And hopefully start to really understand what's going on. Okay, so this is where we ended, with Staffman serving a similar role. What? Okay, that's a repeat. So Staffman binds tubulin subunits. We don't care about that. What we do is very important, but we already said that. Okay, so then we finished talking about the severing proteins. and how they regulate length and behavior of actin filaments and microtubules. And the way you could think of it is they can disassemble, but they can also encourage reassembly by increasing the concentration of subunits available. Okay, and they may play a role in mitosis and meiosis, because you can imagine you're having rapid disassembly and reassembly, so that's very useful if you chop everything up, make all these subunits, recruit them to somewhere else, so that they can set up and do what they need to do, such as form the mitotic spindle or form the, what's it called? What's that thing called? When that cytokinesis? I'm a little sick, so my brain's not working. Huh? The what? The what? Contractile ring. Awesome. You guys can come up and teach today. I used all my energy in recitation. There's none left. Okay. Contractile ring. You have to recruit all that actin to your contractile ring at the right time, in the right place, right? If the contractile ring forms randomly, that'd be bad. So gel Solon, you can imagine, might play a role in that. Okay. So this is all new, starting here. Did you have a question? Oh, stretch. I'm seeing a lot of people stretching today and wanting to go through them. Okay. So now we're going to start talking about proteins that bind to the side of filaments, and they might have a stabilizing or a destabilizing effect. You can imagine how that might work. They might hold things together, or they might break things apart. And so when we talk about microtubules, we talk about MAPs, microtubule-associated proteins. And they are not related to MAP kinases. Someone brought that up today in recitation. I was like, no, they knew that, but I could see how someone might group them together because they're both MAP. But one's microtubule-associated protein, the other is MAP kinases. So totally different. So this picture here is showing MAP localization in an axon and dendrites of a neuron. And so they're standing for two different MAPs. So MAPs are a category of proteins. There are many kinds of MAPs, and they might have different functions. So don't get caught in the trap of thinking MAPs do this. They don't. They do this, this, and this. So one MAP is called tau, and it's labeled green. So it's, yep. And one is called MAP2, and it's labeled orange. And so you can see there are different types of proteins that are functioning in different parts of the neuron and have different functions. Okay, so then there's also MAP proteins that bind to the side of the microtubule, and they're going to project outwards, and they're going to regulate the organization of microtubule bundles. And so you can imagine they're acting kind of like spacers. So here's one called MAP2 that's going to bind to the side and stick outwards. And so all this space here cannot be filled up with another microtubule. So they're all going to be distances, a certain distance. They're all going to be placed a certain distance apart. Tau is another protein that's going to bind, which is associated with Alzheimer's, which is going to bind to microtubules, and it doesn't stick out as far. So those bundles of microtubules are going to be closer together. And so this is a cross section here showing MAP2 and showing tau. And so we are actually looking at each little circle is a microtubule cross section, right? And each of these microtubules has MAP2 bound to it, and so you can see they're spaced kind of equally apart, yet further apart than over here. So these have tau bound to them. And so you can see how these proteins regulate spacing and organization of microtubules in the cell. Okay? Just something to know. So the other thing that's interesting is that MAP proteins are regulated by kinases. Oh, regulation. we thought we were done with kinases. We're not. Remember, kinases phosphorylate things, and they're going to be, that phosphorylation is going to turn things on, activate some kind of interaction, right? So they're controlling the activity and localization of these MAP proteins. So an example is that CDKs, which we're going to learn about in the next unit, cyclin-dependent kinases. These are kinases that regulate the cell cycle, right? Well, a lot of MAP proteins are regulated by CDKs. Kind of makes sense because I've only shown you these two, which have to do with spacing and organization, but also MAP proteins are involved in stabilizing and destabilizing microtubules. So you could imagine, everyone stop and think, everyone pay attention, and then stop and think about why MAP proteins and their role in stabilizing or destabilizing microtubules, why that might be regulated one way or the other in mitosis. Can anyone come just out of nowhere come up with something that might be regulated? Yeah?
Well, when you switching between, I guess, phases in mitosis, so maybe like between anaphase and
Okay, so regulated within mitosis. Well, so something that you should know is that cyclin-dependent kinases tend to increase in activity in the beginning and end of different phases of the cell cycle. So you'd have increased activity at the beginning of mitosis, and then a decrease in activity at the end of mitosis. So you're close, but let's say, what major microtubule structure is involved in mitosis? Okay, what? Right, mitotic spindle. So this is required for mitosis. And so I'm saying some kinase that turns on in the beginning of mitosis phosphorylates and regulates MAP proteins. And MAP proteins affect the structure, the stability of microtubules. It might start to make sense because you're going to need to disassemble some things, you're going to need to reassemble and stabilize some things, right, in making a functional mitotic spindle. If your mitotic spindle is like falling apart, is that going to be useful for segregating chromosomes? No. So things to think about. So all these different pieces I'm telling you about, they all have to do with major cellular processes. And they'll play a role in many of them. So you want to start extending this knowledge into all the things we learn about. Yes?
in terms of like MAP proteins, one of the microtubules, would you say like the microtubules, I don't know, I just keep coming back to that picture that you showed us for that video where like all the microtubules were growing out. And it didn't seem to me like they were growing out like there was so much space between them that they really didn't need like MAP proteins to kind of separate.
Right. So MAP proteins do many things. So let me, okay, so I totally know what you're saying. So you're saying you don't see how these kinds of MAP proteins would be needed, right? Okay. So that's when you're remembering that movie? Is that what you're remembering? Where it was just showing the dynamic instability of microtubules. Okay, is everyone remembering that movie? Where it was they're going in and disassembling and reassembling, right? So yeah, I don't know why you would need this. And in actuality, that movie was made just to show the dynamic instability that is governed by critical concentration. There are no accessory proteins, you don't need anything there. But so these are coming from, I believe, neurons. And so think about the axon of a neuron and how there's probably a ton of microtubules that are organized there because you have a lot of movement of vesicles, right? So in that case, you would need to kind of have an organized network of microtubules. With the mitotic spindle, you wouldn't necessarily need these types of maps, but you might need some other kinds of maps. Did I get there? I guess I'm going to get to them. Wait a minute. Where? Give me a minute. That's actin. So there, I was worried I was leaving something out. Woo! So we'll talk about more MAP proteins, because remember these microtubule associated proteins, and there are some, when you were in recitation today, there were some that are going to bind to the ends and disassemble your microtubule. So they're not spacing, they're actually physically disassembling, and some are going to bind to the ends and stabilize them. And those are ones that would be more likely to be needed in mitosis. Okay, so let's talk about this for a second. So another thing that MAP proteins can do, so remember I said there are many types of MAP proteins. So some MAP proteins can compete with the motor proteins, and so this could regulate the activity of motor proteins. Okay? So if you coat your microtubules with MAP proteins, you're going to kind of block the train tracks, right? So in this, I took this from a paper where they were looking at transport of mitochondria in CHO cells, in Chinese hamster ovary cells. Who knew, right? So here they're showing tubulin and mitochondria. So your tubulin is in green, and so you can see how it's coming out of the center, so this would be an interphase cell, and mitochondria in red. So you can see how there's these speckled dots, and you guys will look at it more closely on the lectures that I post on Blackboard, but there are these speckled dots all over the place. So there's this movement of mitochondria constantly in the cell. Okay. But here, they took the same cells and they expressed high amounts of tau, which is one of these proteins that binds to the side of microtubules. And so if you compare mitochondria in red, notice they're all stuck around in one place, basically in the center. And so if you think about organelles moving and you understand that motor proteins are going to bind to them and walk along microtubules, you can imagine how throwing a bunch of tau into a cell, it's going to bind all over the microtubules and prevent movement of motor proteins. So that's one level of regulation of motor proteins. Obviously, you're not going to express this much tau, and since these proteins are regulated by phosphorylation, you can turn them on and off. You can allow binding, you can inhibit binding, and that could regulate the activity of the motor proteins and movement of organelles or whatever else is being moved by these motor proteins. So this is another MAP protein, MAP2C, and they're overexpressing it. And then also here they're showing that the mitochondria are kind of stuck around the center of the cell, around the nucleus. Over here it's MAP4. It's just showing the blue here is just showing that they're overexpressing it and it's there. And then they're looking at the effect on the mitochondria. And here they're also kind of stuck around the nucleus. They're not able to move. But here, they're co-expressing a kinase, and it's called MARC2. It doesn't matter what it's called. I'm just trying to illustrate the point that this is regulated by phosphorylation. So when you overexpress MARC2, you can, and here you're looking at mitochondria, you can get, well, this is just showing MARC2 and mitochondria. But here, wait a minute, MARC2 mitochondria, YFP. So here they're showing movement again of the, see, they should be showing tau here. Oh, sorry. So all these cells are overexpressing tau. Okay. So when they have tau, which before prevented movement of the mitochondria, when they add in this kinase, so they're overexpressing the kinase that phosphorylates tau, then you recover and you can move again, suggesting that overexpression of a kinase that phosphorylates tau is going to remove it from the microtubules and allow movement of mitochondria again. And so these two are just showing that tau is expressed and MARC2 is expressed. So the reason I showed you this is how people study these things. And so this is the data where they were able to show these proteins, they bind to microtubules, they prevent the function, they prevent the movement of organelles by motor proteins, and phosphorylation of them can remove them from microtubules, allowing motor proteins to move organelles again. Okay? So I'm going to give you guys a minute to look at this and discuss with your friends. And I'm going to circulate, because I want to make sure everyone can kind of understand this a little bit. Because something like this could show up on an exam, I don't know. I might show you data and say, hey, what does this mean? So it's worthwhile. Chat with a friend, take a break, and I'll come around and make sure you guys all understand this. Okay? Don't look scared. We'll do this first and then I'll move forward.
No, she had her slide left for yours.
The same one. It's very opposite. Yep, definitely.
19, we'll have an 18, 19. I was just going around and yeah, 15 months. 15
I'm taking it by the ear. You're saying there's cow. Cow.
You know, I'm positive.
Yeah.
You know how
Yeah, I don't know. They're like, genetics. My arms. My arms. Part 2 is
I got one.
Okay, so it's nice to orient yourself in what you're looking at there. So here you've got your channel segments, so those are control cells that have to finish. It says right here what they're standing for, tubulin, mitochondria, right? And then here, these are these stable cells that are overexpressed in general, where they've got their tau green and so microtubules, just and your mitochondria is red. This is here, what's happening with your mitochondria, which is tau overexpressed. And they did 3 value proteins. You've got tau, you've got MAP2, you've got 4. And every single one of those things have elevated your
Yeah, no.
Okay, guys, how are we feeling? I got a lot of really good questions. Who felt like once they stopped and looked at this and oriented themselves, they were able to get somewhere? Raise your hand. Okay. Who felt like they didn't even know where to start and they wished the withdrawal date had passed? I just saw someone fixing their hair, but I think they really wanted to. Okay. Well, you can always come see me, but my advice with these kind of pictures is always orient yourself first by looking at what you have. So here you have wild type cells. So these are normal cells, and you can look at the normal behavior. And here you have tau overexpressing cells. And then you can, and then I always tell you what you're looking at. Tubulin is green, mitochondria are red. And the whole point is we're looking at movement of organelles along microtubules by motor proteins. And we're talking about how these proteins that bind to the side of microtubules can compete for the space. And that's logical, right? If you stop feeling overwhelmed by all the colors. Color can be overwhelming. And just think for a minute about a train driving on a track. If suddenly someone builds a building on the track, the train's not going to go very far, right? And so that's how you can think of some of these side binding proteins. Okay? Yeah.
Even if the tau blocks it, why would they expect all of the mitochondria to be around the nucleus? Wouldn't there be some mitochondria around like the cell that didn't get blocked? It seems like all of the mitochondria is all connected to the same.
That's an interesting. So the comment was, why are all the mitochondria around the nucleus? Why aren't you seeing some of them stuck in different places? And so they must have. So I guess I would have. That's a really good question. And my thinking is probably that they must have done something to these cells, treated them in some way that put them in some part of the cell cycle or something like that, which is going to be more apt to have the mitochondria in one place so that they can see movement outwards. But I'm not sure, and we have to pull the paper. Yes?
So tau is kind of localizing mitochondria into the nucleus and just stopping them from moving out of where they are.
Yeah, so tau is coding. So if you look, compare these two pictures here. Here, tubulin is in green, and it's how you'd expect, kind of going out in every direction, right? And here, tau is in green. And see how it looks like tubulin? It's totally coating the tubulin. And so the mitochondria are bound to their motor proteins and walking along the microtubules. But when you coat your microtubules with something like tau, it's going to prevent movement. Yep. And so the major point was that if you phosphorylate tau, so compare like this in this corner cell here, which has the yellow, because they're merging the yellow and red here. So this cell got MARC2, but this cell down here didn't. And you can see the mitochondria are all over the cell again when you have MARC2 there, but they're still stuck around the nucleus down here. So that's kind of nice when you see it in the same picture. And so that really drives home the point that phosphorylation of tau is going to cause it to be released, inactivated. Which is the whole point of what I was trying to say, was that they are regulated by kinases. Okay? Okay, I will move on. Feel free, yes?
Wait, so what did the MAP or the tau prevent it from?
They tried three different MAP proteins. Tau, MAP2C, and MAP4. And they all did it. And it kind of makes sense when you consider all MAP proteins are going to bind to microtubules. Right? So they're all kind of acting as a building in the way of the train, on the train track. Okay. Good question. Okay, so let's move on. So we had, let's just do, I'm going to add to the list. Okay, so we're having side binding. I've got MAP. Oh, so for microtubules, you have MAP proteins, and there are many, right? Some have to do with spacing, stabilizing, and destabilizing. And then we just spoke about how they're regulated by kinases. So we can okay, so this is cofilin. And it is a protein that binds to the site of actin and regulates it. It actually destabilizes actin. And so what it does is it binds to the side and twists it even tighter. And that weakens the contact between all the individual subunits and causes it to break. So it disassembles actin. And so what this is showing here is an actin filament, and when you bind, this is the structure of an actin filament, and when you bind cofilin to it, it has a tighter twist, which is destabilizing. But you can kind of imagine if you had like magnets stuck to each other in a chain and you twist them, they'll fall apart, right? Normally. Or maybe not. I guess it depends on the magnets. But I can imagine how you might disrupt those interactions by twisting your chain of magnets. So cofilin binds to the site of actin and it promotes dissociation of ADP-bound actin from the filament. It preferentially binds to ADP-bound actin. And so, therefore, you can make the assumption that it's going to promote the disassembly of older actin filaments. Right? Yes?
Sorry, you just said that it probably binds to ATP.
ADP.
Okay.
ADP, yeah. ADP-bound actin. And so, why? So I'm telling you it binds to ADP-bound actin. How does that relate to the age of actin filament? I'm going to try someone else who I haven't heard from today. I'm going to get my list out and find out who's not here. Janine. Okay, I'll repeat it for you. I don't know if I remember it either. So fast, it's in and it's out, just like that. Okay, cofilin binds to ADP-bound actin, and it's involved with disassembly. And so I'm telling you, it promotes the disassembly of older actin. And so how do those two facts relate? The ADP-bound actin and the age of your actin filament. You can talk to a friend. Chit chat. Yeah.
The one that first comes out with ATP. So
Let's. Do you guys want to call your lifeline down in the front row?
Yeah.
Name Ashley?
don't know if I got it all, but if it's folding the ADP and knocking them off because they're older, well they are older, because they're already what is it, hydrolyzed? Because the new ones come on ATP, so I guess we make them wait for more new ones to come in.
Okay, so she just said the ones that are added are ATP bound, right? And then when ATP hydrolysis happens, it's ATP bound. So if it's binding preferentially to ADP bound, it's got to be older because it had the time for the ATP to be hydrolyzed. You guys are getting that, right? Huh? So cofillin destabilizes the actin filament by twisting it tightly, but it binds to ADP-bound actin filaments, which tend to be older because as the actin monomer is added, you get ATP hydrolysis, right? And so the newer actin is going to be ATP-bound. The older actin is going to be ADP-bound. Yeah.
That cofillin, because that bonds to the whole actin filament, not the monomer. It bonds to the whole filament.
It binds to the filament, not monomer.
What about MAP proteins? Do they bind to the whole filament as well?
MAP proteins bind to microtubules, not to subunits. So the only ones that bind to the subunits, well, I mean there might be more, but that I've taught you, are these ones up here that we spoke about before.
What's the top one here? The map looks at.
Spacing, stabilizing, destabilizing. Those are the major functions of MAPs. Okay, so I'm going to add cofillin over here. What do you guys think? Okay. Any thoughts, questions? Who loves the cytoskeleton? Yay! I see some people raising their hands. That's good. Who likes the cytoskeleton more than importing proteins into the mitochondria? Some people? Okay. In the end, I should take a poll of favorite things you learned. Okay. Yes.
When they bind, is it more like does it have higher affinity towards the end or middle or end or
It binds along the side, and it binds to ADP actin. So as long as it's ADP, it's not a capping protein. It's not. Well, really, that's your only capping proteins tend to stabilize actin anyway. But yeah, it just binds to along the side of ADP bound actin. Yeah. So that question was about where on the actin is it? Along the side. Okay. Tropomyosin stabilizes actin. And so what it does is it binds 7 actin subunits in one filament. So it's a long protein, and it binds 7 at a time. So that kind of makes sense why it would stabilize, right? Because it's kind of acting as a support to hold, it's like a splint, right? That's holding the actin together. So I'm going to add this over here. So cofillin destabilizes. Tropomyosin stabilizes. And so those of you guys who have taken some physiology might be going, oh, muscle contraction. You know this, yay! And so we'll talk about this in more detail later, but that's really, that's a stabilizing side binding protein for actin. Any questions? Okay.
Question.
Oh, sorry.
So is it binding to the ATP?
Does it have a, so you're asking, is there a preference for ATP versus ADP bound?
Yeah.
I don't believe there's a preference for binding to one versus the other, because the binding itself stabilizes the actin. And the reason I say this is because it's involved in muscle contraction, and these are very long-lived actin filaments. So they're going to be ADP bound, right? What do you guys think of that answer? I made it up, but I think it's right. I make it all up, but I make it up based on my prior knowledge that I feel fairly confident about. But I think that's what I think. So the question was, do we think that tropomyosin binds to ATP-bound actin more strongly than ADP-bound actin or anything like that? And so I said, well, based on its role in muscle contraction, it likely doesn't make a difference. And it's going to be ATP. Huh?
The troponin is what would bind to the ATP to move it. So like calcium.
Yeah, that's a whole nother ballgame. We will get there. We will get there, and it's gory detail. Now they're talking muscle contraction. They're getting crazy over there. Okay, let's keep moving. Let's go. Okay, so we got a few practice problems here. Okay, so our first practice problem. Actin filaments in a test tube behave differently than those in a cell. They assemble more readily and have a longer half-life. Why? Huh? Yeah, I think so. I must have. I was looking for extra practice problems. Huh?
I read this like
You probably did. I get all my ideas from the book because I want to get them from places that you can go back and look at. I'm not that creative. I just make you color. Provide examples of mechanisms contributing to this observation. Okay, so since you have seen it before, you have an advantage. Yeah. So I'm going to take a minute to evaluate the first question, and then we'll come back and discuss, okay? You know, I'm saying here, it sounds like a tube, right? But it sounds like a tube based on this question posture. But let's say so guys, so what I'm referring to is they assemble more readily and have a longer half-life in the cell versus the test tube. Okay? So. Sorry, guys. That's what I thought. Yeah. Okay, guys, let's stop for a minute. Let's talk about this, and then we'll move on to the next one. Yes. Yeah.
I was thinking that because in the cell we have monomer binding proteins and the proteins that stabilize the actin, so it has a chance of adding more monomers and staying together than in test tubes.
Okay, so I just heard in the cell you've got these monomer binding proteins that help to elongate actin filaments, and then you've got the stabilizing proteins. So that makes sense. You don't have that in the test tube, you only have actin. Anyone want to add to that? Yes.
I would say that nucleation or assembly more readily due to the ARP complex informants found in the cell that wouldn't be in the test tube.
Right, so nucleation would occur more readily because you've got these nucleating proteins in the cell that don't exist, that you haven't put into the test tube. Right, in the test tube we have actin alone, so assembly and disassembly is governed just by the concentration. That's it. Yeah. Anything else? These are both correct. We're feeling good? Did everyone get that? Did everyone get into the classes they want to get into? Yeah. I see smiling people. No one's Who's taking 409? See, we'll be together forever. Okay. So let's talk about cofillin. Okay, next practice problem that I pried out of the book. Cofillin preferentially binds to ADP actin. And this is totally what we just talked about, but I'm hoping you guys can just pull it right back out. Cofillin preferentially binds to ADP actin and is therefore more likely to disassemble older rather than newer filaments. Why? So I'm not going to let you talk about this because we just talked about it. Do you want to talk? Are you going to talk about? Are you going to talk about this, or are you going to talk about dinner? Okay. So talk for a minute in case you didn't get what we spoke about before, and then I will come back in like 2 minutes. We did, but you know, not everyone. Okay, guys. So what was the answer here? Cofillin. Most of you know this, but for those of you who actually just figured it out in the last 2 minutes while talking about classes and dinner, cofillin preferentially binds to ADP actin and is therefore more likely to disassemble older rather than newer actin filaments. Why? Why? Oh, come on. Yes.
ADP is older and more apt to disassociate.
ADP is older, more apt to dissociate. Well, it's older, so it's going to bind to ADP. Yep.
The cap that it has from taking longer to turn from the
Oh, but this is actin.
It's not too bad either.
GTP cap, there's I guess the GTP cap is more having to do with microtubules and with actin. Yes, there's stronger binding with ATP bound actin. But continue your thought.
Oh, I was just going to say that because it's still in the process of changing, that it's going to be bound stronger to what it's connected to, and so it's going to go from the bottom that's already been assembled for a while and no longer in the ATP state.
So it's going to disassemble the hydrolyzed. Yep.
Yeah, that's what I was just gonna be like, the newer ones is ATP, so
Right.
cofillin.
Right, so newer actin polymer is going to be ATP bound, and older actin polymer is going to be ADP bound. So that's how cofillin discriminates.
I missed that.
Okay, Okay, proteins that bind to the ends of filaments have greater effects on filament dynamics than proteins that bind to the side. So we just talked about all these side binding proteins, and we're going to talk a little bit about some more side binding proteins that are going to affect stability and stuff like that. But then there's these end binding proteins. Specifically with actin, there's CAPZ, which caps at the plus end of actin, and it's stabilizing. And so if you look over here, so this, you guys have seen this kind of graph before, and you'll see it in this week's recitation practice problems, where you're seeing your growth of your actin filament, and based on your concentration, right? So this is your critical concentration, your steady state. And so your uncapped actin filament is going to grow like this, and your critical concentration is going to be here. But when you add a capping protein, which is going to stabilize it, so you're not going to have any more growth, but you're not really going to have any disassembly either, then it's actually going to change your critical concentration, making it greater, so it's actually less likely to disassemble. And so basically what you have is you've got this stabilizing effect. You have stabilizing, and you also don't have any growth either because you've capped the end. So one molecule of capping protein has greater effects than one molecule of side binding proteins. Why? I don't repeat this. Never. One molecule of capping protein has greater effects than one molecule of side binding protein. So compare CAMZ to tau, or not tau, but to all these MAP proteins that could stabilize or destabilize. Not even MAP. How about compare it to a molecule of tropomyosin? Okay. Yeah.
So like the tropomyosin it binds with 7 active proteins at a time, but the cap, like once you put the cap on, there's no like growth or disassembly. So you can still have disassembly if the tropomyosin is only binding 7 at a time.
Right, so tropomyosin only binds 7 at a time. You can still have disassembly elsewhere if it's not bound by tropomyosin. Cap is gonna, that's where things are coming or going. Yes.
Because it binds to the side, you still got the ends open. So as you said, it binds 7 at a time, like those 7 you can still add on more to the end or take off some from other end. But once you put the cap on, you can't stick nothing else to it.
Right, so tropomyosin just binds to the side, but your ends are still open and available. So you can either add to or leave from still, but once you add your cap, you can't add or leave at all from your ends. Yes?
I have a quick question. Just for the plus end, is that I'm assuming it's like the enzymes or like the protein?
No, you just add your plus and minus ends of your actin polymer, of your actin filament. Yeah. Yeah.
So is it only at the plus end?
Well, CAPZ caps at the plus end. If in your muscle, your minus end is held by what? Who remembers? Tropomodulin. Is that right? Who said that? Is that right? I can't remember off the top of my head. I think I even teach you. Yeah. So, so you do have capping at both ends in muscle, and that's why these actin filaments are so long-lived. Who just asked me that? Okay. Not my day today. Okay. Anyone else? Anyone else have questions, thoughts? Okay. Everyone having fun? Yeah.
So when there is one cap, are we assuming there's another cap?
No, it depends. It depends. No. So just because there's one cap, don't assume there's another cap. If you cap your plus end, you might. It depends on what your actin's doing, because you're not only in muscle, right? You're also moving forward. You're also making contractile rings. It depends on the function of what needs to happen, what proteins are going to be recruited. Yes.
I'm just asking to just get the complete stabilization of it, because can't stuff add and come off from minus end, but just at a slow rate. So if it's capped at the plus end, does that mean mean, it's like nothing even coming from the myosin, or it's just like much lower than usual?
Yeah, so, okay, so it depends on what's being recruited. So if you only have CAP-Z recruited, yes, you can still be leaving from your minus N, and you just won't be growing at your plus N. I'm not sure. We have to think about what purpose that would serve. But in muscle cells, then you're going to have both ends capped because you need them to just be stable. So it just depends on what proteins are being recruited. And so we'll go through different functions, different processes, and the different kinds of proteins that got involved. Yes? Yes.
You said tropomyosin probably on 7
That tropomyosin binds along the side and binds 7 subunits at a time. Cap only binds one, but it binds the end. And that's where your coming and going is happening. And so that's why it has a greater effect. Okay, no. Okay, yes.
So when you're trying to think of examples of like CAP-Z use in muscle cells.
That's one. I'm wondering if I have any better examples coming up. We're going to see because I think I have a better example coming, but I can't think of it off the top of my head right now.
So I was gonna ask like where we would see this, but since you just said muscle cells.
Well, muscle cells is one.
Okay.
Yeah, muscle cells. Okay. Are we good to move on for a minute? Okay. So I guess I should add here capping. This really slows me down. Is it worth it? Yes. Okay. So this is just kind of a comparison of the effects of proteins on filaments, and it's dependent upon stoichiometry in a way. So for one tropomyosin, that covers 7 active subunits. 1 tau will bind to 4 tubulin subunits. 1 cofilin will bind to 1 actin subunit. Capping proteins, you can get 1 CAP-Z for 1 whole actin filament. So that's why it has such a huge effect. All you need is 1 CAP-Z. So end-binding proteins are way more effective than side-binding proteins. Okay, what do you think about it? Oh, there you go. Tropomodulin at the minus N in muscle cells. Right, so that's what we were just talking about. In muscle cells, you have this long-lived actin filaments, and they're capped at the plus N by CAP-Z and at the minus N by tropomodulin. I don't need to ask you how it affects the dynamics because we just spoke about that. Okay, so then there are these proteins. Actually, let's just add tropomodulin. CAP-Z with plus N. Tropomodulin minus N for actin. Okay, so then there are these other MAP proteins I keep talking about. Remember I said there are ones that are involved in spacing and some that are involved in stabilizing and destabilizing. So these are proteins that are MAP proteins that bind to microtubule ends. They're not capping proteins per se. So you can have some MAP proteins involved in stabilizing. So it suppresses catastrophes even though your GTP will be hydrolyzed. And so this would result in long, less dynamic microtubules. And then you have MAP proteins such as catastrophe factor, kinesin 13, which is not a kinesin. And it's destabilizing, and it increases the frequency of catastrophes. So you have shorter, more dynamic microtubules. And so there's this kind of competition between the two of them. And then the other thing to think about is regulation. These different MAP proteins are regulated by phosphorylation. So during mitosis, for example, this XMAP215 that's involved in stabilizing microtubules will be phosphorylated and inhibited. Do you want, it depends on this, you might say, wait a minute, do you want stable or unstable microtubules during mitosis? Well, it depends on where you are in mitosis. Because you have this period of time where you have highly dynamic microtubules because you're reaching out trying to find your chromosomes. And then there's this highly dynamic period where you're segregating your chromosomes. Well, that's actually, you're going to probably activate a catastrophe factor then, I guess. Okay? So I guess I'll add these probably over here. So with the maps, so you can have, what did we say, X map 215? X map 215, stabilizing. These are examples of maps. And then you'll have kinesin 13 or just catastrophe factors. And the catastrophe factor, is that stabilizing or destabilizing? Yes. Huh? I'm putting it, so it binds to the end, but I don't like to call it a capping protein because capping proteins imply stability. And these are not necessarily stabilizing, but they are MAP proteins because they bind to the side. They bind to microtubules. And MAP stands for microtubule associated protein. Okay? Any questions about this? No. Good. Okay, so then there are these plus tips. And so these are plus end tracking proteins, which are also MAP proteins. And they bind to the plus ends of microtubules. And then they dissociate when microtubules shrink, which is kind of cool if you want to do live cell imaging and follow the growth of your microtubules, isn't it? So catastrophe factors and XMAP 215 are both plus tips that modulate growth and shrinkage. And then there are other plus tips, such as EB1, and that controls microtubule positioning, and it's essential for yeast mitotic spindle positioning. Okay, so there's just a whole bunch of proteins that bind to microtubules and affect their functions. That's what it comes down to. How are we doing? Okay, another few minutes. Okay, so in order for the cytoskeletal filaments to be useful, they need to be organized and attached to the cell structure, right? If you just have like an actin filament floating in the cytoplasm, it's not very useful. Well, I guess it can be, but usually it's attached to some kind of cell structure. Same with your microtubules, right? They're attached to your centrosome. That's kind of like an anchor, and they come out, they radiate out from there. So let's just go back to intermediate filaments for a minute. I feel the need to give them a little more attention. So these, remember, this is this bundle of tetrameric subunits, and it's important for giving the cell strength and resistance to pressure, and organizes cells into tissues, right? And so they're held together by different lateral contacts between each other, and that they also have accessory proteins that do this, so to help to organize the intermediate filaments. So one here showing plectin, which is in green, and it's a cross-linking protein that functions in bundling intermediate filaments, which are in blue, and linking the intermediate filaments to microtubules, actin, myosin, and the plasma membrane. So, do you think, so based on the importance of plectin in regulating and organizing intermediate filaments and cross-linking them to other cytoskeletal proteins in the cell, Do you think an individual with mutations in the plectin gene would be viable? What do you guys think? Yeah, no. What would happen if your intermediate filaments were not organized in any way or attached to anything? They wouldn't do anything. And how would your cells respond to pressure? Yeah. How would your skin be in this nice structure, or would your epithelial cells attach properly to your basement membrane? Would your tissues organize into tissues? No. So this would probably be bad, I would say. It's pretty horrible, actually. They have a little section in the book you should read about people who have a mutation in this gene. They're subject to like neurodegenerative diseases and it's in skin conditions, and it's a bad situation. And mice with mutations, I think knockout mice that are knocked out for plectin, die, I think, in either early after birth or embryonic. I forget. So it's important. Accessory protein. Okay. Actin filaments. So we've been talking about all these accessory proteins that are regulating growth and shrinking of actin and microtubules, and this is all really important. So actin filaments are organized into bundles and web-like or gel-like networks, and that has to do with their function. So we're starting to transition over to function a little bit. So foramins make bundles. So these stress fibers, contractile bundles, or these tight parallel bundles that are associated with filipodium, these kind of protrusions. I take that back. But ARC complex makes webs, this kind of gel-like network, which is more associated with the leading edge of the cell and movement forward. So when you think about the contractile ring, you should be thinking foramins. And when you think about the leading edge of the cell, you should be thinking ARC. But it's important to organize all this actin, because remember, ARC's going to nucleate, and foramins are going to nucleate, and you'll get growth of your actin polymer. But these actin filaments need to organize in some way so that they can function. Yeah.
Is this the same foramin that profilin interacts with?
Mm-hmm. This is the same foramin that profilin interacts with. Yep. So we're kind of going back and taking these little bits and pieces and starting to assemble them into some functional thing.
I thought it built the actin chain.
Foramins nucleate actin, but in certain conformations. And ARC nucleates actin in different conformations. Remember, ARC binds at an angle.
Yeah.
It binds to the side of another. So you start to have this kind of web-like conformation with ARC. With foramins, it's just this linear growth. Okay, so this all kind of comes together with you have these cross-linking proteins that are going to organize your actin polymers into whatever structures that are needed for the purpose. So you've got these bundling proteins and gel-forming proteins that stabilize and maintain actin structures. And so this is just some examples. The red part is the actin binding site, and so you can imagine that here you would bind to actin along here and over here, and that would cross-link and stabilize an actin structure. Filament might work differently and create a different kind of structure. So they're cross-linking actin so bundling proteins are cross-linking into actin filaments into a parallel array, and they help to determine what proteins interact with the actin filament and can determine function. Okay, I'm gonna stop here for today, because everyone's looking a little stale, including myself. And we will continue this excitement on Wednesday. Let me just shut this off real quick.